All right, we are on chapter 19. So this is the last chapter in Unit 1, and it's on the cardiovascular system, specifically blood vessels. So blood vessels are the delivery system of dynamic structures that begins and ends at the heart. They're going to work with the lymphatic system to circulate fluids. And so the blood vessels are going to be things like our arteries, our capillaries, our veins. Arteries are the blood vessels that carry blood away from the heart. Typically, the blood is going to be oxygenated except for pulmonary circulation and the umbilical vessels of the fetus. Then we have capillaries, and these are in direct contact with the cells. And what they do is they're going to deliver the oxygen, the nutrients, they're directly serving cellular needs. And then we have veins, which are going to carry blood back toward the heart. And typically it's going to be carrying deoxygenated blood, except for pulmonary circulation and the umbilical vessels of the fetus. So this would be systemic circulation. We start off with the heart. Then we have arteries, and you can see it branches into smaller and smaller arteries until we get all the way down to terminal arterioles, which will go into capillary beds. This is where oxygen and nutrients are going to be exchanged. And then this is going to drain back into a postcapillary venule, back up into larger and larger veins, back to the heart. In addition, we have the lymphatic system that's also going to pick up fluid around capillary beds and bring it back and kind of dump it into the veins, and it'll go back into the heart. All vessels consist of a lumen and a wall. So the wall is going to be surrounding the lumen, and the lumen is the part that contains the blood. And then the walls of all vessels except capillaries have 3 layers or tunics. There's the tunica intima, the tunica media, the tunica externa. Now, capillaries have an endothelium with a sparse basal lamina. The innermost layer that is actually in contact with the blood is called the tunica intima. And this is where we talk about that endothelium, that simple squamous epithelium tissue that lines the vessels, is continuous with the endocardium, and provides that slick surface which helps to reduce friction. And then in vessels that are larger than 1 millimeter, there is also a subendothelial layer, which is just connective tissue basement membrane. Then there's the middle layer, the tunica media, which is composed mostly of smooth muscle and some sheets of elastin. And then we have sympathetic vasomotor nerve fibers innervate this layer, and this is going to control vasoconstriction and vasodilation. And vasoconstriction is a decrease in lumen diameter, and vasodilation is an increase in lumen diameter. This is the bulkiest layer responsible for maintaining blood flow and blood pressure. The outermost wall or layer is called the tunica externa. It's also sometimes called the tunica adventitia. I will continue to call it the tunica externa. It's composed mostly of loose collagen fibers that are going to help to protect and reinforce the wall, as well as anchor it to surrounding structures. Now, in larger vessels, there's sometimes found something called the vasa varum, which is a system of tiny blood vessels that function to nourish that outermost external layer. So if we just look, we have the tunica intima, endothelium, subendothelial layer, and then don't worry about the internal elastic membrane. But you can see both arteries and veins have the endothelium and the subendothelial layer. We have the tunica media, smooth muscle and elastic fibers, thick tunica media. Not so thick here. And then we have the tunica externa, collagen fibers, somewhat I mean, it's not as thick as the tunica media in arteries, but it is thicker. And then it is quite thick in veins. And then we also have the vasa varum, which are the blood vessels that you can see inside here that are actually nourishing this external layer. And then we would have capillaries with these endothelial cells and the basement membrane. This table is just showing you, we'll actually come back to this table later, but we have arteries, we have different types of arteries, elastic, muscular, and arterioles, and then the walls consist of different types of tissue. So with elastic arteries, we have more smooth muscle, some elastic tissue, but with muscular arteries, we have a lot of smooth muscle, and also a good bit of fibrous tissue. And then still smooth muscle is found in the arterioles. And the reason I'm pointing this out is because some of these are going to play a role in vasoconstriction. Two of them are. And one of them, elastic arteries don't play much of a role in vasoconstriction, but they do have smooth muscle. Muscular arteries do play a really important role in vasoconstriction, and you can see they have a lot of smooth muscle. And then arterioles will also vasoconstrict as well. Capillaries, again, only contain the endothelial layer. And then we have venules, which have very little elastic tissue, a little bit of smooth muscle, some fibrous. And then veins, which have some smooth muscle and some fibrous. And then arteries are divided into three groups. They're based on their size and their function. We have elastic arteries, muscular arteries, and arterioles. Elastic arteries are the thick one, large arteries. They have a low resistant lumen, and the examples given are the aorta and its major branches. They're also called conducting arteries because they do conduct blood from the heart to medium-sized vessels. Elastin is found in all three tunics, mostly the tunica media, and like I pointed out on the last slide, or 2 slides ago, they do contain a good bit of smooth muscle, but they are inactive in vasoconstriction. They do play a role as pressure reservoirs that expand and recoil as blood is ejected from the heart. And this is important because it allows for continuous blood flow downstream, even between heartbeats. The elastic arteries give rise to the muscular arteries. These are called distributing arteries because they deliver blood to body organs. Most of the name arteries are muscular arteries. They have a thick tunica media with a lot of smooth muscle but less elastic tissue, and they are very active in vasoconstriction. Then we have arterioles, which are the smallest of all arteries, and they have, the larger arterioles have all three tunics, but the smaller arterioles just have that single layer of smooth muscle around the endothelial cells, and they control flow into capillary beds by either vasodilating or vasoconstricting. And they're also called resistance arteries because they change diameters. Because changing diameters changes resistance to blood flow. And they do lead to capillary beds. That's where we start talking about capillaries. Capillaries are microscopic vessels. Their diameter is so small that only a single red blood cell can pass through at a time. The walls are just a thin tunica intima. And they also have these things called pericytes, which help stabilize the capillary walls, control permeability, and they also play a role in vessel repair. And capillaries supply almost every cell in the body except for the stuff that we've talked about in AMP1 that's avascular: cartilage, epithelial tissue, the cornea, the lens of the eye. Capillaries, their job is gas exchange, oxygen exchange, carbon dioxide exchange, nutrients, waste, hormones, all that stuff between blood and the interstitial fluid. Now, all capillary endothelial cells are joined by tight junctions with gaps called intercellular clefts. And this is all capillary endothelial cells, okay? And they're going to allow the passage of fluids and small solutes, which is important because that's what capillaries do. They allow things to leave, to go out into the interstitial fluid to where the oxygen and the nutrients and all that kind of good stuff can get to the cells. There are 3 different types of capillaries. There are continuous capillaries, and these are abundant in skin, muscles, lungs, and the central nervous system. I do want to point out that the ones found particularly in the brain are unique. And this is because they form the blood-brain barrier, which is a totally enclosed capillary with tight junctions and no intercellular clefts. I don't know if you guys remember from, again, AMP1, the blood-brain barrier, you had the astrocytes with the perivascular feet that are wrapping around, and we talked about the tight junctions, and it's going to allow very little to pass through. So these would be your continuous capillaries. This would be a pericyte, endothelial cell right here, and then this intercellular cleft. And the tight junction, again, is where they're kind of hooked together to where things aren't going to want to pass through there. But here we do have a place for stuff to pass through. There's fenestrated capillaries. These are going to be found in areas involved in active filtration, like the kidneys, or absorption, like the intestine, or endocrine hormone secretion. The endothelial cells contain these pores that almost look like Swiss cheese holes, Swiss cheese-like holes, and they're called fenestrations. And what they do is they increase permeability. And the fenestrations are usually covered with a thin glycoprotein diaphragm, but that plays very little role in what goes in and out. So we have the fenestrations are these larger, right? We still have the intercellular clefts, but we have these larger pores, these fenestrations here and here, here and here. And then finally we have sinusoidal capillaries, fewer tight junctions, usually fenestrated with larger intercellular clefts, incomplete basement membrane, usually have larger lumens. These are going to allow stuff to pass through, right? This is going to allow cells to pass through because of how large the holes are in the capillaries. I'm about to show you a diagram of it. But they're found in the liver, bone marrow, spleen, in the adrenal medulla. These capillaries are very winding and the blood flow is sluggish. And this is going to allow time for modification of larger molecules and blood cells that pass between the blood and tissue. And then in addition, these type of capillaries have macrophages that will capture and destroy foreign invaders. So it almost looks like somebody was making a capillary and just gave up, or I don't know, but there's still capillary here, right? There's still a wall here. It's just got a lot of places for stuff to pass through. So large intercellular cleft, there is still tight junction, incomplete basement membrane. Yeah. And they're going to be winding. The blood flow is going to be sluggish. They're going to contain macrophages. All right, capillary beds. Interwoven network of capillaries between arterioles and venules. And then we talk about microcirculation, which is the flow of blood through a bed from an arteriole to a venule. A terminal arteriole branches off of an arteriole, and then that terminal arteriole will branch into 10 to 20 more capillaries that will form the capillary bed. And that's where the exchanges of gases and nutrients and waste and all that kind of good stuff takes place. The capillaries then will drain into a post-capillary venule. And then flow through a capillary bed is controlled by the diameter of the terminal arteriole and upstream arterioles. In addition, local chemical conditions and arterial vasomotor nerve fibers regulate the amount of blood entering a capillary bed. So what they do is, if blood is needed, the arteriole and the terminal arteriole will dilate. If blood is not needed, they will constrict and it'll shunt blood away. So here we have arteriole, terminal arteriole, and blood is flowing through here. But if we vasoconstrict here and here, blood will no longer pass through this capillary bed and it'll be shunted somewhere else. Capillaries found in serous membranes of intestinal mesenteries do have two additional features. They have a vascular shunt and a precapillary sphincter. Capillary shunt is a channel that directly connects the arteriole with the venule. It's actually going to bypass the true capillaries. It consists of a medarteriole and a thoroughfare channel. And then there's the precapillary sphincter, and this is just like this little cuff of smooth muscle that surrounds each true capillary that branches off the meta-arteriole, and it just literally acts as a valve regulating whether or not blood is going to flow through there or not. And it's controlled by local chemical conditions. It's not innervated. So first, let's look at this. We have, this is the meta-arteriole, so this can literally just go right through. But in addition, we have these precapillary sphincters, these little cuffs of smooth muscle. And if these cuffs of smooth muscle clamp down, blood does not flow through the capillary bed. Instead, it bypasses the capillary bed and goes back to the venule. Veins carry blood toward the heart. The formation begins when capillary beds unite in postcapillary venules and then finally merge into larger and larger veins. So the capillaries do unite to form postcapillary venules, and it's going to consist of endothelium, a few pericytes. It's very porous, and this is going to allow fluids and white blood cells into tissues. And then larger venules have one or two layers of smooth muscle cells. Veins are formed when venules converge. They have all three tunics, but thinner walls with large lumens compared with corresponding arteries. The tunica media is thin, but the tunica externa is thick. It does contain collagen fibers and elastic networks. So the tunica media is thinner because it doesn't contain as much smooth muscle. And then we have the large lumen and thin walls make veins good storage vessels. They're also called capacitance vessels because they contain up to 65% of the blood supply. And this is just showing an artery, nice thick walled, strong structure, and then the vein, a little bit thinner walled. The lumens are a pretty similar size here, but typically veins would have a little bit bigger lumen. And then this is just showing that 60% of blood is found in systemic veins and venules. And then veins, the blood pressure is lower in veins than it is in arteries. And so there are adaptations to help ensure the return of blood to the heart. So large diameter lumens offer little resistance, so there's little resistance to blood flow. And then there's also valves, and valves are going to prevent backflow of blood, and these are going to be found pretty abundantly in the veins of limbs. And then there's venous sinuses, which are flattened veins with extremely thin walls, composed only of endothelium, and the examples given here are the coronary sinus of the heart and the dural sinuses of the brain. So again, tunica intima, similar. Thinner tunica media here with the veins, yes. Thicker with the arteries. Tunica externa is thicker on a vein. And then again, this table is the same table we've already seen. So if you remember, the muscular arteries play a role in vasoconstriction. Again, they got a good bit of smooth muscle, but so do elastic arteries, and they really don't play a role in vasoconstriction. So again, capillaries, only endothelium. Venules, not a lot. There's some endothelium, some smooth muscle, some fibrous tissue. And with the veins, we have some smooth muscle and more fibrous tissue. Last thing we talk about are the anastomoses. They're vascular anastomoses, or interconnections of blood vessels. Arterial anastomoses will provide alternative or alternate pathways that will ensure continuous flow of blood, even if one artery is blocked. These are common in joints, abdominal organs, the brain, the heart. However, none are going to be found in the retina, kidneys, or spleen. And then there's arterial venous anastomoses, and these are shunts and capillaries. And the example given is the medarterial thoroughfare channel that we talked about. And then there's venous anastomoses, which are so abundant that occluded veins rarely block blood flow. Here are your questions. And that is all I have for part 1.